All right, good morning. Today we're going to talk about DNA replication. I hope you guys sort of understood the structure of DNA and especially how the nitrogen bases pair up. It's very important to notice that the A's and T's match up and the C's and G's match up. All right, that's going to help you a lot when you're looking at DNA replication. All right, so in this class, we're not going to get into a bunch of depth. We're just going to look at the basic processes and only learn about 3 enzymes. I think you're only going to have to know 1 on the test, but I mentioned 3 of them. There's other things that go on in DNA replication, but we're not going to cover it in high school. All right, so the first thing is, what is DNA replication? Just remember from the last unit, before cells can go through mitosis or the first stage of meiosis, they have to copy their DNA or copy their chromosomes. So this is what they do when they're doing that. All right, this is DNA replication. This is how they copy their DNA to get ready for cell division. This is a good diagram here. The blue represents the original or the parent strand, and the pink is the daughter strands. So this one strand gets unzipped and eventually gets turned into 2 different strands. All right, so the process, there's 2 key enzymes that are involved in the process. We're going to mention another one in a second, but there's 2 key ones. The first one is the DNA polymerase. A lot of times you see it called polymerase 2 or polymerase 3. You just need to know DNA polymerase. But this is what brings over the other nitrogen bases that are floating around in the nucleus to pair up with the unzipped DNA, okay? They just sort of help get the right nitrogen base to the one that's already there. All right, DNA helicase unzips the 2 strands. And the next example is going to make a lot more sense to you. All right, but when they pair up, you just need to make sure A's and T's go together and C's and G's go together. And this is a good diagram here. You can sort of see that this is the original strand. It's getting unzipped by the helicase, and the polymerase is bringing over these new strands to join to make the new strands. All right, so replication. All right, there's the place where the 2 strands get divided by the helicase. It's called the replication fork. Just a vocabulary word. Just know what the fork that means. That's mentioned. You can sort of see the replication fork's growing. It's going to keep going, and this thing's going to keep getting unzipped. You guys do not need to know about the 5 and 3 ends and all that type of stuff. All right, I did mention a third important one. It's called the DNA ligase. This is what helps create this covalent bonds on the DNA backbone on like the sides of the ladder, really. Remember, like those things have the fish where the phosphates and sugars are at? This is what helps combine or link those together. There's 3 main steps of replication, and these words all are great use of the words to help describe this, but initiation, elongation, and termination. Initiation means to start. This is when helicase unzips breaks its hydrogen bonds really between the nitrogen bases and gets it ready to start getting new DNA added to it, all right, new nucleotides added to it. Elongation, making something longer, that's when new DNA strands grow 1 base at a time. So the existing strand that got split apart, got unzipped, is the template for the new strand. So when you have that new strand, then now you have the double-sided double helix. And then termination, that's when another enzyme cuts it off, but now you have 2 new double helixes that have replaced the original helix. DNA replication is called semi-conservative. It's a term that's used to describe what it is. And the reason we use that is because each helix that is created contains 1 strand from the original helix, and 1 is a new strand. So the replication of one helix results in 2 new daughter helixes, or helixes, and each one contains 1 of the original parent strands. So you can sort of see this is a really good overall summary of what replication is. Sort of see here's where the helicase comes in, starts unzipping it. There's a DNA polymerase helping move the new things. Don't worry about what Okazaki fragments are. You don't need to know those. You don't need to know what RNA primers are. Here's that ligase back here helping bond these things together. So everything's going good there.
Excuse the interruption.
This right here is a good YouTube recap of DNA replication. I would highly suggest you watch this video. All right, it's a really good recap of DNA replication. But the big things to take away is what initiation is when they're getting unzipped. Elongation, the new nitrogen bases are being added to create that second strand to make a daughter strand. And knowing what termination is. So this here is a good example. This is the best picture, I think. Shows what's going on. This blue is the original strand. Helicase is here, unzipping it. The polymerase is bringing these new bases to join, to pair up, to complement each other. That's what it's called, complementary strands. And it's going to end up making, when it eventually gets to the termination end, it's going to have 2 new strands of DNA. And it's called semi-conservative because one of the strands, like one of the sides of the DNA, is from the original strand, and the pink strand is the ones that got created to make the new strand. Right?